Alright, gang, we are at video number 3, or video C for section 16.2. This morning, Mr. Ethan here, say hi. He's not going to say hi, but anyway, Ethan decided to join me. He woke up a little bit early. You say hi? Hi, there you go. And hopefully he doesn't touch the screen while I do this, but anyway, yeah, so he's going to join us. We are picking up, this is the super detailed section on DNA replication. And it's what I'd like you to do. There's a lot to take in on this one, but if you can just, there's a whole lot of proteins involved in DNA replication, and if you can just kind of keep in mind the beauty that's in the complexity, and just it is an absolutely amazing system for something that happens every single time a cell wants to divide. All these proteins all working together beautifully, intricately. It's an amazing situation. Put that into the confines of a creator, or put that into the confines of evolution, whichever way you're coming from, and it is it's mind-blowing. It really is. So hopefully you can get some appreciation on how that's going there. Anyway, but the okay, where we left off yesterday, or with the last video, all we did was describe DNA polymerase III and how it always builds molecules of DNA in the 5' to 3' direction. It only ever adds to the 3' side. And so we end up with a leading strand and a lagging strand. And if that's confusing, just go back to the last video. But the some things that we missed out on. If you listen to the last video, or as you were reading, we talked about the DNA polymerase III having the limitation of only adding to the 3' end of an already existing DNA strand. And so that actually, I just kind of said that in passing in the last video. What that sets up is a situation where DNA polymerase III, it can't actually start laying down new DNA until we've already put down some DNA. It can't actually start from scratch putting DNA down. So what we have to do, and what cells do, is they make something called a primer, and that's this thing right here. And a primer is just a short segment of either DNA or RNA that is first laid down on the template strand, so that DNA polymerase III, or in the future other things, can start to add nucleotides to it. Okay, and so here we have a primer. When it comes to replication of DNA, the primer is actually made out of RNA, which is very similar to DNA, just a different sugar. Anyway, it's just chemically a little bit different than DNA. And the molecule, this pink molecule here, is called primase. I have it highlighted here. It's a protein you need to know. And so primase comes along, lays down a primer. Now we have a starting spot. Just a quick note here, we are working on the lagging strand. Here is what we'd be working on, the lagging strand. So it would be unzipping in this direction, and we're going to put a primer down, fill back in this direction, and then later on there'd be a primer up here and fill back in this direction, and so on. But anyway, so this primer laid. Now that we have the primer laid, the RNA primer, now DNA polymerase III, which is this molecule right here, DNA polymerase III can attach and starts to build build a DNA strand from the 3' end of that RNA strand. Okay, so it can build on from there. So as it continues to go, we have a problem when it's done. So here we're showing DNA polymerase III detaching. When it's done, we have this primer here. Okay, so DNA polymerase III detaches. It's going ahead to wherever a primer would have been laid here, and it's going to build another strand of DNA up until it runs into this primer. Okay, so we built one section. We're continuing to unzip in this direction, and there's a primer out there. Anyway, so here the DNA polymerase III is again. Ethan, you're in the way, bud. And it's building a DNA, this next Okazaki fragment, until it gets to the primer, and then it's going to detach. And we've got a problem. We have RNA in the middle of our DNA. Okay, so what's going to happen is a new polymerase called DNA polymerase I is going to go through, and its job is to replace the primer. So when this gets up to the primer, here we have DNA polymerase I is going to go one nucleotide at a time, take out that RNA nucleotide, replace it with a DNA nucleotide, take out the next one, replace it, take out the next one, replace it. So you can see that happening right here. Once we've replaced the entire thing, DNA polymerase I will detach, And now we would have, you can see it right here, a little gap between this Okazaki fragment and this Okazaki fragment. And so I had mentioned this yesterday, but ligase, DNA ligase down here, and I should have highlighted that, but DNA ligase is going to fill in that gap. And then we have completed that. So that's kind of the details on the laying of the DNA, and we have to start with the primer, add DNA onto the primer because of the limitations of DNA polymerase 3, and then we have to replace the primer, it's DNA polymerase 1, and then close the gap between the DNA strands. Okay, so that's kind of the details of the building of the DNA. We're going to take one more step in on detail because there's some other proteins that are involved in this entire thing that we want to identify, and then that's as detailed as we're finally going to get on this whole thing. So, all right, here is the super detailed diagram of DNA replication, and just to kind of give it a where are we in the whole process here. This section of DNA that we're looking at, this is the replication fork. That if this is our replication bubble, we're on this side that's in the box here, okay? So we have our leading strand on top that just continuously, as it unzips, follows along. We have our lagging strand on the bottom that had to start and work backwards, start and work backwards, start and work backwards, so on. The leading strand, we're just going to kind of show some things, but the lagging strand here, we actually show kind of sequentially starting on this side. This would be the first strand, second strand, and so on. But anyway, extra molecules, we had said the DNA unzips, okay? The DNA unzipping is caused by the green molecule in the front here. And let me change my pen, and Ethan's trying to make himself more comfortable here. The green molecule in the front here is called helicase, and I want to highlight that. Helicase is one that you have to know, and that's the one that does the unzipping, okay? Another thing to remember is that this unzipping, it's not only unzipping, it's also unwinding, and so we have a helicase, a double helix, and we're unspinning that double helix. And if you've ever had two strings that were kind of wound together, you can even try this. Take two shoelaces, wind them together, tie the end of them, the one end, and then grab the two shoelaces and at the other end that aren't tied together and pull them apart. Pull them apart. If you pull them apart, you'll notice that it's pushing the twist towards the other end, and it creates a strain. Okay, so there's another molecule that is not mentioned on this slide, but you'll need to know it, called topoisomerase or topoisomerase, and that molecule releases the strain that would be building out ahead of this whole thing as we're unwinding the double helix. It releases that strain. The book doesn't talk about how, but basically it'll snip, let it unspin, and then put it back together. But anyway, that's topoisomerase. Excuse me, my eyes bothering me. Anyway, so helicase does the unwinding and the unzipping. Once a DNA molecule is unzipped, the two single strands that lead away from that, they will very, very automatically want to join back together. And so we have to have something holding those apart. So we get something called a single-stranded binding protein, or a series of single-stranded binding proteins, okay? And those are these gray things right here, and they're basically holding the strand open so that they don't just automatically rezip together, because they are attracted to each other. The hydrogen bonds will pull them back together, okay? So that is single-stranded binding proteins. We can say DNA polymerase 3 up here on the leading strand, and it's just zipping along. I've got questions in previous years about, is there a primer somewhere on the leading strand? And yes, there is. If I look back at this diagram, that little red spot is the primer all the way back at the origin, and then that primer was what started the entire process here, moving that way. So that's the leading strand, nothing big there. The lagging strand is where we see kind of the sequence of things. We have primase laying a primer here, okay, on this Okazaki fragment. The previous Okazaki fragment already has the primer and DNA polymerase 3 building backwards. Here in the previous one before that, we had the primer plus a completed section of DNA, and we actually have DNA polymerase 1 replacing the previous primer. And then all the way back here where that's all happened, we have a ligase that's closing up the gap. Okay, but this is a great diagram to study and figure out. There's a bunch of steps shown here. All the molecules that you need to know are shown here, with the exception of that topoisomerase or topoisomerase. So that leaves that there. I'm pretty much done with this video. It was a short one. I just wanted to verbally be able to express this diagram. What I would recommend now and I'm going to put this on the website, but there is a YouTube video called, and let me make sure I have the name right, and again I'm going to put it on the website. It is called Molecular Visualizations of DNA, okay, and it shows an absolutely fabulous DNA replication video. You would go to, if you go to the 1 minute and 41 seconds time on that, 1 minute and 41 seconds, it shows DNA replication, and what it shows is that this diagram is really a bad representation. Okay, now this is easy to understand somewhat, but the right representation, which you'll see on that video, is that all of these proteins, the helicase, the binding proteins, the RNA polymerase 1 and 2, and ligase, all of these different, and the primase, anyway, all these things, they're actually all attached into one big complex, and then the DNA zips through that complex. The video describes it. It'll blow your mind. It'll absolutely blow your mind that that is going on inside our body. Anyway, so enjoy that video. Again, it's the minute and 41 second point of the molecular visualizations of DNA. It'll be on the website, or you could look it up on YouTube. It depends on what you want to do there. But anyway, I think that's it. We have one more section coming from section 16.2, so we'll have a 16.D that talks about mutations, and we'll leave it at that. Have a good day.